So good afternoon, everyone. I hope everyone had a wonderful lunch and we are all ready to learn and listen from the speaker this afternoon. So I am Rosemary Deacon and I will be the moderator for this afternoon session of the second day of the SINSM event of the biology department. So without further ado, let me introduce to you our speaker for this afternoon. So, Sir Arvin Jetmar Kaida is currently an assistant professor in the Department of Biology, College of Science here in the University of the Philippines, Baguio. He graduated with a bachelor's degree in science with a major in biology at the University of Santo Tomas. This was where his love for science started. His first exposure to research was during his thesis, where he conducted research on fish parasites in Lake Palakpakin in San Pablo, Laguna. So here, he developed an interest in the field of parasitology and taxonomy. He then pursued his graduate studies as a DOST scholar in UST, where he graduated cum laude with a master's degree in biological science. His thesis and research was about the conservation and parasites of the endemic crocodile, Crocodylus midorensis in Palawan. In this study, he also received the research grant of IUCN SSC Crocodile Specialist Group. So after getting his master's, he decided to practice his degrees and enter the academe, where he taught senior high school students at his alma mater for 1 year. He then decided to take the leap and pursue his PhD in Japan, where he entered the program of environmental science at the University of Shiga Prefecture in Shiga, Japan, under the MEXT scholarship. So his dissertation was about the biogeography of parasites from bufo species or frogs in mainland China. His main works deal with the biodiversity, ecology, and conservation of both the host and their parasites. So specifically, he's inclined in the taxonomy and systematics of these parasites using both traditional and modern practices or modern approaches. He was also a member of the Philippine Society of Parasitology from 2017 to 2018 and the Japanese Society of Parasitology from 2019 to 2021. He has also published a number of his studies under well-known and accredited journals in his field. His studies were also presented in both national and international conferences. So currently, he is also a consultant to the NRCP project, which studies host-parasite interactions in Cagayan River. He is also a project staff under the NRCP project, which studies snail parasites in Batanes. So as a returning scientist, he aims to establish a career in both academics and research by conducting more biodiversity studies about host parasites and its interaction. So very impressive yung next speaker natin for today. So before we start the lecture, may I remind everyone that while the lecture is ongoing, you can already type in your questions in the chat box. So let us all give a warm round of virtual applause to Dr. Jetmar Kaida, who will share his lecture on bioinformatics entitled Back to Bank, Basic Phylogeny Using the GenBank Web Tool.
Okay, thank you very much, Ma'am Rose. Thank you for that wonderful introduction. So, hi everyone, good afternoon. Magandang tanghali or hapon po sa inyong lahat. Andito po tayo ngayon sa SINSM to learn many things. So, one of it is my presentation for today's meeting. So, I'll share it now. Can everyone see my presentation screen? Okay, thank you very much. So today we'll talk about back-to-bank basic phylogeny using GenBank tool. So again, good afternoons and welcome to the 28th SINSM. And our main theme is about the modern and flexible teaching, and its sub-theme is about the biodiversity information using in our digital age. So since I'm talking about we're talking about digital age and biodiversity information, so I decided to present to you, of course, bioinformatics. So just to give you a brief overview of what we will talk about. So first we'll have the introduction, then GenBank and its intro, features, website layout, and format. And then I will give you a brief summary or discussion about my study, which is on phylogenetics. And lastly, we'll have the demo or tutorial on how to use the GenBank this afternoon. So to give off, so when we hear bioinformatics, I think most of you will think of uh, of course, this following words. So we have DNA genomics, we have computer biotechnology, genetics, evolution, information, and database, and barcoding and such. So in database, so this is very important in bioinformatics. So in perspective of biologists, when we say data archives or database, so this is what we usually think of. So we have here, of course, the sets of our samples. And of course, this is just for fun. This is how movies portray the data archives for biologists. But for bioinformaticians, so the data archives looks like this. So mainly computers, hard drives, mainframes, such as that. But they have similar perspectives. So most of them are in bulk groups, and they are contained in a big room, and they are, of course, big data. Okay. So now, what do we expect from a database? So when we talk about database, these are the followings that we should expect. So in bioinformatics, of course, so it should be a sequence, functional, structural information, and related bibliography with well-structured and indexed, and it must be cross-referenced with other database and periodically updated. And of course, without tools for analysis and visualization, this would not complete the bioinformatics. So next, so what is bioinformatics? To define it, so it is the use of computer to store or retrieve or analyze and or predict the composition or structure of biomolecules. So we'll tackle about this further in the next slides. So it aims to develop the database containing all biological information, and it is applied in different aspects such as evolutionary studies, medicine technology or medical technology, gene therapy, forensics, and many more. Now, it's important is to store and manage biological data and information in computer readable forms both by humans and the computer. Now, bioinformatics uses both primary and secondary database. So what are these primary and secondary database? So first off, we have the primary database, which is a original submission or an archival. So this means that all the data comes from laboratories or scientists, and they are only in sequence or structural information. Sequences such as nucleotide sequences, which is managed by NCBI, DDBJ, and EMBL, and also protein sequences database, which is managed by Swissport, PIR, and GenePep. Now, secondary databases are both human and computer curated. So yung primary natin, these are now checked and curated by human and computer, and this is used to derive analysis or treatment of primary data. Now, they are also protein structure databases managed by PDB, EBI, MSD, MMDB, and also we have the domain and motif database managed by ProCite, Blocks, and COG. Now, to go to the main focus of our topic, so we have now the GenBank. Now, to have a general grasp about the bioinformatics, so let's talk more about GenBank. So GenBank was created in 1979, and it was first called as the Los Alamos Sequence Database, but it was renamed GenBank in 1982, and it was under the National Library of Medicine at National Institute of Health. And it is part of National Center for Biotechnology Information, where it is collaborated with International Sequence Database Collaboration. So this branch of the NIH or National Library of Medicine aims to establish a public database where the research is in computational biology and to develop softwares, tools for sequence analysis, and of course to disseminate biomedical information. Okay. So GenBank is an annotated collection of all publicly available nucleotide sequences and their protein translation. Publicly, it means that they are open access, so everyone can access all this data. And currently, it is one of the fastest growing repositories that receives the sequence produced in laboratories throughout the world from more than 100,000 distinct organisms. So there are three types of or branches of databases that are collaborated by INSDC, or the International Nucleotide Sequence Database. So these are your NCBI GenBank, the DNA Data Bank of Japan, or DDBJ, and we have lastly, of course, your European Molecular Biology Laboratory, or EMBL. Now, we have here the statistics for the GenBank and the WGS. So this shows how GenBank and WGS are increasing rapidly. So as we can see here that from its establishment during the 90s, it has grown into several nucleotide and bases, and of course the WGS, or the whole genomic sequences or databases. Okay. And as we can see here in the statistics, so they started in December 3, 1982, where they only receive 606 sequences. And as they further accept sequences, it grew until, of course, millions and billions of sequences with, of course, trillion bases. Okay. So by the way, WGS started during WGS, or the whole genome shotgun sequences, started during April 2002. Okay. Now, in GenBank features, there are different submission types and submission tools. So for submission types, we have the direct submission, where scientists or laboratories submit their nucleotide sequences in traditional or standard records. We also have the complete microbial genome, the whole genome shotgun, or the WGS sequences, the high throughput genome sequences, and of course the bulk or the batch submission, the EST, GSS, and STS. But for today's discussion, we'll only focus on the direct submission. Now you can submit your sequences through BankIt, Submission Portal, Sequin, and TBL2 ASN tools. So I will proceed to the flat file structure. Now this database, or the GenBank database, contains only ASC text file or flat file and FASTA file. Now I will explain this further in the next slides. So here we can see the GenBank website. So we have the GenBank information. Here you can submit your nucleotide sequences, genomes, WGS, metagenomes, TPA, TSA, NSDN, of course, their documentations and other researches. So later in the workshop, we will discuss how to use this website. Now for GenBank records, so we have the traditional record. So that consists of the three main parts. We have the header. Sorry, I'll just use the pointer. So we have the header, the feature table, and the sequences. So the header consists mainly of the flat file or text files, while your sequences consist of the nucleotide bases. Okay. Now for traditional records, so here we have the whole flat file or the header. So we have here locus, definition, accession, version, keywords, source, reference, etc., etc. So we will divide this one by one. So for the first, we have the locus, which contains 6-character accessions, which is usually the first letter of the genus. So yung A, or for this one, we have 8-character accessories or accessions, should I say. So this contains the first letter of the genus or the species names, and followed by the number of the submission. So that is the accession or the locus name. And then we have the base pairs or the number of nucleotides. And then we have the type of submission or the nucleotide. We have here the mRNA. And lastly, we have the division. So this shows the division, which is invertebrates, and of course the date of the submission. Yan, ito pala yan. So yan. Okay. or modification date. Next is we have the identifier. So here, if we're going to look for some nucleotide sequences, we have to also focus our search here. So here we have the accession, which is a unique number assigned to the piece of DNA when it is first submitted. And this is the usually the one that is found in the publication. So this is cited whenever we see or look at the publication and they put these sequences here. And it has two formats, of course, the 2 letters and 5 digits, or 1 letter or 6 digits. So the version number and gene index number, or the GI, is to identify the current version of the sequence. So here it has been modified 2 times. So if this is 3, so it has been modified 3 times. So yun. Next is we have the organism. So here we can see kung saan galing yung DNA or RNA na or nucleotide sequences natin. So here, for example, we have the source, the species name, and of course, its common name, and of course, yung NCBI's taxonomy. So andito na yung from yung classification niya. So yan. So if we are looking for organisms, so we should focus our search here. Okay. Next is we have the featured table. So here we can see, or it includes annotation information about the gene and the gene products, as well as the regions with identifiers and qualifiers. So the source fields, So the source fields, this one, it provides the length of the sequence, scientific name, of course, and the taxonomy identification number, this one. And optional informations includes the clone source or the tissue type of the cell type. Okay, so this one, tissue type, it came from the lateral eye of the Limulus polyphemus. Okay, then for the CTS, okay, so this is the gene field. This part is about the nucleotide coding sequence and its name. For DNA entries, of course, there's the CDS field, and which is information about the boundaries of the sequence that can be translated into amino acids. So this one. So this is already the translation of the nucleotides into amino acid. So we also have the product, myosin 3, the protein ID, of course, and the codon start. Okay. So that is the featured table. So this is usually from GenPep's ID. Next is we have the FASTA format. This is the important to look at when we want to find our nucleotide sequences. So FASTA format, it is called FASTA because it must contains the definition line and of course the nucleotide sequences. So for the definition line, we have the GI number and the database identifier. So these are usually the database or gen bank that where we deposited the nucleotides. So for example, we have here GB or gen bank, and or EMBL for European Molecular Biology Laboratory, or DDBJ, or database of Japan. Then we have the accession number. So ito yung sinasabi ko kanina, 1 letter and 6 digits. And we have the locus name. Okay. Now, to give you an example of how to conduct or use this GenBank or bioinformations in a study. So I present to you my study, which was my dissertation during my PhD. This is entitled Phylogeography of Parasitic Nematodes Recovered from Bufo species in mainland Japan. Okay. So the focus of this study is on biodiversity, biogeography, characterization, and evolutionary relationship of nematode species from Bufo of mainland Japan. So I did both morphological and molecular characterization, which help in species identification and establish their possible genetic groups. Now, before I was only conducting biodiversity through traditional model, but now I did a molecular approach, which is, of course, bioinformatics, and it serves as supplemental tools in better understanding diversity and taxonomy. Okay. Now, the site was, of course, Japan, mainland Japan. So Japan is divided into 3 regions. So you have your Hokkaido, mainland Honshu, and of course the Okinawa region. So where I did my study was of course in Shiga, in this part in the center of the Japan, and I collected samples of bufo from different regions of mainland Japan. Okay, so we have the Kanto area, Kansai, and the Kyushu. Okay, so these are the types of bufo that we collected. So we have Bufo japonicus, Bufo japonicus formosus, and we also have Bufo torrenticula. And we also collected different species or genus of frogs or amphibians such as Ranidae and Rhacophoridae, and also salamanders, which is Salamandridae and Hyenobidae. Okay. Okay, so the method of this study was to conduct sampling in different region and collect toads, and we dissected them and of course observe parasites using dissecting microscope and collect the parasites in vials with 90% ethanol. Now for morphological studies, so of course we mount them in slides using glycerin water or lactophenol, and we identify them using microscopes. Of course, we use nematode keys. And for SEM, we've conducted this in order to have a better look at the nematodes. Okay. Now for the important part, which is the molecular methodology. So we collect the tissues from the nematodes and use alkaline lysis to extract DNA. And of course, we perform PCR, and we submitted the sequences to the FastMac database, and they sent us the sequences, and we aligned and analyzed them using the Finch TV and Mega version 10. Okay. Now the focus of this study is all about the family Rabiaceae, which is discovered by Rylate in 1915. They are called lungworms because they usually parasitize the lungs of both amphibians and reptiles. So the main focus of the species focus of this study is the Rabies species, which is the Rabies incerta. Okay, so this is the line drawings of the Rabies and the SEM of the Rabies incerta. So they are widely distributed globally, excluding Antarctica. And the difficult thing about this study is that there are less morphological variation. That's why a traditional method or morphological studies is difficult to assess this species group. And in Japan, they only have 9 species. So this study resulted to the following or creating of the phylogenetic tree of these species, nematode species. So using the 28S region, so these are all samples from my study. So we collected their tissues and of course made of course 28S nucleotide sequences from them, and we conducted a phylogenetic study. And here we observed that all of my or this confirmed that all of my samples are belong to the Rabiaceae family, while we have the out group Cosmocercidae, and we have of course the related the Strongyloididae or Aloinematidae. Okay. Now to further look at the Rabies species, so here we have the main phylogenetic tree. So let's just focus on the Rabies. So here we collected all the Rabies species from the bufo or the toads. So we were able to separate them according to different clades. So the clade 1A, which belongs to the Kansai region. The clade 1B is only in Nara. The clade 1C can be found only in Kyoto region. And clade 1D is in and clade 1E is are only found in the Kyushu or Kanto region or the northern part. Okay, and then we were also able to found a new possible new species and cryptic species. So we have the Rabies species 2, which can be found in Oita, and we have the we were able to generate a sequence from Rabies tokyoensis, which is also new, and of course our new possible species also. Here we have Rabies species 1, and they are clade into 1 clusters. Then we have of course different genus species of Rabies found in different species of frogs. So they belong to Anurans and Rana family and Rhacophoridae groups. Okay. Next is we have, of course, we use biogeography for this, and we were able to determine their location. So we were able to notice that, of course, the blue ones are, or the clade 1A, B, C, and D, E. So they are all in mainland Honshu, and they are separated by their hosts. Okay, and the green ones are from different anuran groups such as the Rana and Rhacophoridae, and the red ones are from salamanders. Okay, so this is what phylogenetic study looks like. And lastly, we were able to compare our samples across the world. So we were able to determine that, of course, the Rabies family in Japan, which is or the Rabies incerta, are very far from the different species of Rabies in different places or different countries. So only the Rabies tokyoensis and cafunata and the Rabies species are related to other Rabies across the other countries. Okay. So the findings of this study, of course, is that the lineage identification using mitochondrial gene is more efficient for the genus Rabies because the gene mitochondrial is more specific for identifying species level parasites. And the Rabies species has a wide host range which parasitize amphibians, reptiles of various orders and families. So in parasites, they can be considered specialists or relative generalists. Now, the Rabies species group could be tracking the host resources instead of host phylogenies. Thus, the cross-transmission could occur in nature by overlapping or sharing of habitats. And since bufos are terrestrial and semi-aquatic, so there's a chance that, of course, the Rabies can transfer from different hosts. Okay, and of course, there are physiological aspects such as epidermal properties or defense mechanisms of the host, which allows the nematodes to enter to its skin easily. Okay, or more difficulty. Sorry. And then, of course, there's a evolutionary host switching that is happening in the Rabies species group. Now, Rabies incerta exhibited a higher level of variation in this gene due to the following. We have the difference in the host segregation because in Japan, the bufo host is segregated by region and geographical distances, which makes the isolation or the gene isolation even more distinct. Okay. Now, in Japan, the distribution of the subclades of R incerta, which we have showed to you that they follow the same pattern as bufo species. Okay, and the divergence or the separation diverge into eastern western groups through the formation of or an expansion of ancient basins in Japan. So this makes it this makes the isolation of both the host and the nematodes or parasites. So it exhibited great genetic variation due to reproductive isolation, yet they shared similar genetic structure due to cophylogeny. So it is evidence that the host parasites cophylogeny and population level dispersal events. So yeah, so thus the parapatric distribution pattern of closely related species or intraspecific clades of Rabies strongly suggests that the diversion of Rabies species through geographic isolation is rather than host specification. Okay, so if you want to read further into this study, so this study was published in a Parasitology International, and of course, to wrap things up, this bioinformatics. So one mentor said to me that we cannot study something we do not know. So it's important to have informations about what we want to study, and that database is very important in the field of science because without database we cannot, of course, do a lot of research and studies. And bioinformatics is still growing, but it's serving its purpose in collecting data such as nucleotide and protein sequences. Now, GenBank is very useful in identifying organisms and its lineage or evolutionary history, biogeography, and genetic variation, or in terms of phylogenetic, of course. So that ends the lecture for this session, and we'll continue with the workshop later on. So thank you very much.
So thank you very much, Prof. Jet, for that interesting and very informative lecture po. So guys, alam na natin yung gagawin pag gusto natin mag-Japan, mag-PhD lang tayo. Okay, so there were many points that were brought up in the lecture. So let me just summarize a few. So first, many biological information are available in both the primary and secondary databases, and these information can be used in a variety of fields. So sa case nung kay Prof. Jet kanina, environmental studies yung sa kanya. So apart from that, as mentioned sa introduction, we can also use bioinformatics techniques and medical technology, forensics, and more. So second point na we also learned how to read traditional records found in GenBank, which allows us to correctly identify which information is relevant to our needs or our research. And last point that I would like to add, so yung use ng molecular techniques along with the morphological techniques for species identification is especially important here in the Philippines, since we all know that the Philippines is very rich or very rich in nature. And then alam natin na marami pang species or organisms that remain unidentified. So hopefully this lecture will inspire you to conduct more species identification studies here in the Philippines. So ayun, before we proceed with the demonstration portion of Prof. Jet's presentation, let us first have our open forum. So you may now place your questions, reactions in the Q&A section here in the Zoom. And please do not forget to indicate your name and affiliation together with your question. So yan po, let's wait for a few questions. So while the other participants are still formulating and typing their questions, allow me to start with one question. So Sir Jet, in your PhD study po, you were the first to generate the DNA sequences from 2 nematode species there in Japan. So in case I want to conduct a similar study here in the Philippines, what do you think are the obstacles that I would encounter if I do conduct a similar study here? So yun po.
Okay, that's a very interesting questions, Ma'am Rose. So first thing is that, of course, this is very interesting study to conduct in Philippines. So, however, of course, we don't have the enough capacity to conduct larger or bigger phylogenetic studies in Philippines because, of course, we lack we only have one sequencing company, or the Philippine Genomics. I think it's better if we can first start with basic phylogeny, and we should agree in using, of course, the same primers in identifying species or taxonomic, of course, in microbiome or other fields. So I think it's better if we start simple, and yeah, I think that's it. Thank you very much.
Thank you very much, sir. Second question naman po. Since na-mention kanina na ang use ng molecular techniques is for species ID, what other types of studies kaya yung pwedeng i-conduct after na ID na yung species? So parang on the application part naman po of research yung question po.
So aside from taxonomy or systematics, of course, we can also conduct environmental modeling using different, of course, genome or of the species, or Yeah, and we also can see we can also observe the evolutionary lineages or history of this species, and of course there's what we call the gene flow. So it's also important to study this, and this will help in conservation studies. So I think that's one use of molecular studies aside from taxonomy or systematics, and also we can also use this for epidemiology or of course medical aspects. So we have of course this was booming in during the pandemic, since the COVID was studied through the use of molecular approach or bioinformatics. So there are lots of studies about COVID that uses bioinformatics and molecular approach. Okay.
So thank you, sir, for answering that question. So we can all see na ang daming applications ng bioinformatics today. And I hope na we all get interested, or we all got interested into the topic. So one more question, I think. Since marami sa participants natin ay nagtuturo ng senior high school students, how do you think this topic would be introduced, or how should it be introduced to younger people, since based sa files pa lang medyo nakaka-overwhelm po. So para yung itsura po parang comsci kasi as compared sa bio po. So yun po.
Yeah, so for of course for educators, so I think we can use this as a source or to conduct meta-analysis or any how to say data-driven studies. Of course, in GenBank, we have lots of sequences there, and we can do many things using its nucleotide sequences. So I think we can use it to teach students to first, of course, to know about different studies such as this, like phylogenetics. So we can start from there, and we can learn the relationship between animals or the sequences that we aim to study. So for example, we can conduct a phylogenetic between different species of birds or frogs, something like that. So it will be interesting for students to know their relationship, and of course their lineage or history, evolutionary history. Okay, so yan lang. Thank you very much.
Thank you din po, sir. So ayun po, for younger students, or yes, younger pupils, based sa answer ni Sir Jet, it would be best to give examples ng animals, or kung may students din pong mahiling sa plants, so pwede rin pong mag-focus sa plants, since applicable naman po both on animals and plants ang bioinformatics or molecular techniques. So ayun po, may pahabol po bang question from the other participants? Kindly chat na lang po. Okay, I think nasagot naman lahat ni Sir Jet yung tatlong questions. So thank you very much, guys, for the questions, and then thank you very much for listening as well. Of course, thank you then, Sir Jet, for that lecture. And then before we start with the workshop proper, let's have a 15-minute break before we proceed. But a few reminders. Let me remind the participants to download and install the Mega application, which will be used later in the workshop. So, i-take na natin tong opportunity na we have an expert with us para malaman natin kung paano talaga gamitin yung phylogeny, or paano gawin yung phylogeny techniques. So, ayun guys, let's come back ng 2:30. So, ayun po. Thank you very much po.
Thank you.
is awarded to Assistant Professor Arvin Jet B. Marcaida for his valuable input in the 28th SINSM Biology Cluster with the theme Facilitating the Collation and Interpretation of Biodiversity Information in the Digital Age, held via online webinar platform this 28th day of April 2023. Signed by Professor Radi J. Remillo, SINSM Committee Chairperson, and Professor Dimfna N. Javier, Dean of the College of Science. So again, thank you very much. That was the last talk and workshop for the BioSysNet portion. So this concludes the 28th SINSM of the Biology Cluster. Again, thank you very much for all the participants, and we hope that you enjoyed and learned a lot from all the lectures and workshops. So please be reminded that participants should answer the evaluation form again, and we would like to express our, to move on to the closing portion, allow me to welcome our dear department chair here in UP Bio, Professor Ronnie J. Kalugay po for his closing remarks.
On na ba ako? Hello, good afternoon po. So, maraming salamat po in behalf of the Department of the Biology, College of Science. We thank you for attending this online 28 SINSM. So, mabuti po nag-online tayo. Medyo meron pong mga cases dito sa campus. So, some classrooms were evacuated and disinfected just today and the other day. So, maraming salamat po sa inyong pagdalo at sana we were able to achieve our goal of sharing with you the knowledge and training that we had, at sana it will just spread like fire, most especially to our students. So as scientists, as researchers, as teachers, I hope this event has enriched us all. So, noong ako po ay isang young na guro, ay ang gusto ko po ay pumasok sa pahinungod system ng UP system. Yun, ang pahinungod ay nagpupunta sila sa iba't ibang liblib na parte ng Pilipinas para makapagturo sa mga estudyanteng hindi na nagdarahop na hindi na walang access sa education. Hindi natuloy yon, pero I realize na I am in the right place in my life right now with regards with that goal. Na where you are now is exactly where you should be actually. Marami po tayong mga hinahandle na estudyante right there in the classroom. You are impacting lives. You are impacting the hope of the nation, and that is our students. And I hope that through this event, we have enriched and honed your skills, and ultimately you hone and mold all these young minds that day in and day out. Siguro sometimes we take it for granted na napaka-routine na lang ng trabaho natin. Hirap-hirap magturo, ang hirap-hirap pumasok, pero each day, each subject, each student, lalo na yung mga nasa likod, sometimes nakakalimutan natin sila. At yung mga nasa harap, di ba, yung sila yung magagaling at nagpa-participate. Pero for me, I made it a point na every student will come away with something. And I hope na yung mga natutunan natin ngayon it will refresh us. Ano ba talaga ang mission natin as teachers? So that ordinary routine every day as teachers, as researchers, ang dami po na tayong nai-impact na mga buhay na mga young lives na yan. So maraming salamat po sa inyong pagdalo, and I would like to thank the Department of Biology SINSM team who have put this this event together for so many months. Marami pong, parang simple lang siya, pero marami pong brainstorming na nangyari for us to come together this week. And of course, I'd like to thank our speakers from Dr. Jet, Professor Serrano, Professor Soriano, Sir Dison, Sir Paul Ignacio. Sino nang speaker natin the first kahapon? Ay, hindi ko rin maalala. Sorry ho, hindi ko maalala sa dami ng ginagawa. Ang UP Systems Network Office, Sir Don and friends, salamat po. That's why very smooth ang ating online event. To our research assistant, Ms. Vera Sabas, Ms. Stephanie Terao, Ms. J. Antalyad, at ang ating, di ba, nagagandahan po ang ating mga moderators. Yan, si Rosemary Quinomes, si Ma'am Jodeline Melegrito, si Ma'am Chelsea Bagni, si Ma'am Alan G. Marquez. Lahat po yan ay mga full-time na mga guro ang ating mga moderators. May nakalimutan ba ko, Ma'am Ross? Ma'am Kauthar, yan, napaka, at saka si Ma'am Kimberly Paglingayen. Mga full-time teachers po yung mga yan na hindi po sila basta-basta very passionate din po sila sa pagtuturo and sa pagre-research po. Kaya alam po nilang ginagawa nila sa pagmo-moderate. And of course, I would like to thank the head of the Department of Biology. I will not let this finish without hearing from him. First, from his deputy, Ma'am Alan G. Marquez.
Hi, sir.
Ma'am Alan G., can you say something? Yan.
Hello,
Yan po
sir.
ang pangalawang punong abala.
Yes po. So, first
Ma'am
of all,
Allen, wala kang sound.
ay, wala po ba?
Narinig ko.
Ay, naririnig.
Okay,
Hindi
sige.
ko naririnig. Ay,
Okay.
wala
So,
pala akong okay. Ayan, go.
okay. Yan. So, first of all po, I would like to thank all the participants who went through the rigors of securing a slot, no, from pre-registration up until emailing us frequently, no, para magbayad dun sa ating registration. And ayun po. So, maraming salamat po sa pag-participate ninyo actively. So, naging interactive po yung mga talks natin and very maximum po yung participation nila. So, with that po, I'm very grateful. And also po yung SNO, as always, yung technical support nila and yung ating tech team. So, RAs Vera and Steph, and sa lahat po ng moderators and speakers. So without all of you, this event would not be possible. So I'm very grateful po. So yun, in behalf of Sir Jeno na rin po, we are very grateful. So yun po. Thank you so much.
Ayan. And now, thank you, Ma'am Allen, sa mga pahirap nyo. And of course, let's hear from the department point person ng SINSM, Mr. Jeno Ray Pagaduan. Sir Jeno, nandyan siya? Ay, wala po siya. So anyway, ang ating palang first speaker, di po ba, napaka-interesting ng ating first speaker, si Dr. Aris Reginaldo, yung nag-talk on Philippine vertebrates. Amazing talk. And he's a very prolific scientist. So mabuti po at nag-attend kayo nito. Our speakers are well selected. So, again, maraming salamat po. At ito po ang inyong lingkod, the Department of Biology, College of Science, University of the Philippines. God bless you all po. Happy long weekend po, kaya ganyan ang background ko. We can all now relax. And before we sign off, let's have a group picture po. Can you turn on your videos po? Ma'am Rose, take it away.
So good afternoon po. Good afternoon po. For those participants that are able to, kindly open po your videos for a photo op po. Yes po.
Yung mga co-host din po, pwede mag-promote ng attendees. Thank you.
All right, sir.
Sige po.
So, promote. Mm.
Inaayos lang po ni Ma'am Rose. May konting technical.
Currently at letter J.
I-open lang po ang mga cameras nyo for our group picture. I-open po.
Done po. Paki-accept na lang po yung panelist na invite. Thank you. Sa mga attendees po, paki-raise na lang po ng hands yung mga gusto pang mag-open ng camera. Thank you.